Hello everybody. Today I will be showing how to edit workflows in the geneXplain platform. Let's log in geneXplain platform. This is one of my logins. Here we have a standard view of the platform, and we go to the data and we see our demo project. Today we are building a workflow to analyze gene expression data and DNA methylation data together. So first let's load the data. So we will load the data into the data folder of the demo account. We'll load them from our computer. So first is the transcriptomic data and the DNA methylation data. So they will be loaded as two different tables. So first let's import the transcriptomics data. So it starts from the first line. The data begins at the second line. And these are gene symbols. Okay, and then we import the DNA methylation data, which is in this table. So here we are. So you have two tables. One is contains almost 400,000 lines for methylation accession numbers, and another contains 20,000 genes with their expression values. So okay, so then we can open this folder, we see these two tables, and the first what we are going to do is to create a workflow. So for that we click right button and select here new workflow. Let's give a name for the workflow, for instance Transcriptomics and CGmethylation to transcription factor binding site. So it creates an empty screen where we can put our workflow. First we need the entries for input data. So these are green. This will be created with this green button. So we click here. So we first create an entry for the transcriptomics data. So this will be the data element, and the default value we put as a project URL, and the role is input, and the type of element actually table of reference type of unspecified, because we can put their transcriptomics data in any format. So then we create another entry for the methylation. And again, this will be data element. It will be here in the project. It will be input, and it will be table, no, not text, table, and also unspecified type. So we have these two entries. So now first we want to actually convert the transcriptomics table into the internal ensemble format. So for that we would put here a method called table conversion. And this method will be actually converting unspecified, basically auto to gene ensemble. And we need to connect the input, and we have to create the output. So the output will be the table with the name of Transcriptomics ensemble. So this will be again the data element, and now we can give a default name as transcriptomic, and we add here to the name ensemble. So it will have the new name of the file will have this ensemble ending. Okay, so we should connect the output. Sorry, we have to delete this. So this output, we have to connect it, this output, to this table, and we need actually a second table which will have the not matched entries in case if they are not matched. So we can actually make it auto temporal, and let's make it this name, and we can give not matched. Okay, so and then we can connect this to this. The same we have to, okay, so we don't need to convert in CG methylation data, it's in standard format. So the second what we want to do is actually to create to analyze this data and to find differentially expressed genes, and we will do it using lemma. So we have a statistical package lemma. We'll put it here. So, and it will get as input this table. The same we will do it for DNA methylation site. So we need lemma again, and we connect the input. So we need also the output folder, so the lemma output data into the folder. So we are adding a folder for the lemma output. For that we click on this button. We say that's lemma for transcriptomics. Just it will be actually a folder, a data element. Yes, it's a data element, right? And we took the name from the transcriptomics ensemble, and we add here lemma. So now we can connect this output here. So in the same way we did it for the lemma for CG methylation. Yes, you can see we select the data element and put this name. Okay, so now we have to specify more exactly the lemma. So for that we click on lemma method, and we see many lines here, many options. So first we have to specify that it is unnormalized values. Then we have to specify which conditions will be selected for tumor and which conditions will be selected for the control. So we have to give to the workflow the possibility to select conditions. So for that we actually then put here the input values. So let's call them conditions for transcriptomic tumor. And this will be multiple strings which will be selected from the table columns, numerical only, and we specify which table we choose. We actually look at the table which describes the transcriptomics. And this option then we put here. So and then we have to add this option to the lemma. So we select the line here, which has to be connected to those options. So with this button, we connect it. Okay. Then the same way we create conditions for the control. So these are conditions for transcriptomics control. And here we have the multiple strings and taking from the table numerical columns, and the table which we choose here again, the transcriptomics, and then these conditions which has to be selected for the control. So we can connect this to the control. So now lemma is ready. So let's save this workflow. Now let's specify lemma for the CG methylation. Same way we will add now different conditions. So let's add conditions CG tumor. This is multiple string taken from the table, and the table now is actually CG methylation table. Okay, let's connect it again. Tumor connected. Now conditions for the control. Control for CG control. So this is a multiple string and taking from the table columns, and table will be CG methylation. Ready. So we have to connect controls to these conditions. So they are ready. We save the workflow. Now let's have a look what kind of input mask we had. So if we click on this button, we see how this workflow look like. So when we can specify tables by drag and drop here, and then we can see which conditions we can select for the transcriptomics and for the methylation. So here we put methylation, here we have transcriptomics data, and then we can select multiple conditions by pressing these buttons for the tumor and for the control. But the only problem here, I see that the order of those input values is not optimal, so we should probably reorganize it a little bit. So let's edit this workflow a bit. So what we want to do, we want to actually put the option for transcriptomics on the first place, and the CG methylation should be actually place number 4. Right, and then we in place number 2 we'll put the conditions for tumor and for the control of transcriptomics, place number 3. And now, and for methylation, we will put here, so 3, 4, 5. And 6. So now, if we save it and start, you see it's arranged a little bit better. So we have first transcriptomics data, so we can put here. And then we have we can put here the methylation data, and then we can select here for tumor. For instance, we know that tumor is these 2 conditions, and control is actually these 2 conditions. And for the methylation, tumor is actually these two conditions, and the control is these two conditions. So now it's ready to run, so we can try to run it as it is. You see, it's actually now converting the table and initiating the lemma. So we'll wait until it will finish. So as you can see, the result, the calculation have just finished, and it saved the result in these different folders. So we can see the new folders. The folder with ensemble, lemma for the transcriptomics data, tumor versus control, and the results for the tumor versus control for the methylation. So if we click on such table, for example, for transcriptomics, we see a table of 18,000 genes. Here we have accession numbers from the ensemble for these genes, and then we have these log fold changes calculated by the lemma, so we can sort by these fold changes. You can see these downregulated genes with a minus log fold change, or upregulated genes from the plus, and it calculated the p-value and the adjusted p-value, so we can actually see which genes have significant p-values. Okay, so we can return to this workflow and work on it further on. So now, as we can see that this workflow produces these two tables. So correspondingly, tumor versus control for the transcriptomics and for the methylation data. So now next, I want to actually to select the most upregulated or downregulated genes according to the statistical criteria. For instance, for the tumor, for the transcriptomics, then we would add here a method for the selection. So filter table. So we can connect this table to this filter table method. Oh no, wait. It has to be done from here to here. Sorry, we have to delete it. Yeah. So we connect it from this to this, and then we'll select what we are going to do in this filter table. So we would like to add here the filter conditions. And let's have a look what kind of conditions we want. For instance, if we return to this table and we want to filter it by log fold change which goes above a certain threshold, log fold change above, let's say 0.7, and p-value below 0.5, so we can actually check how many genes we can choose by these conditions. So then if it's good enough, so we have 1,900 genes. So it's a good set of genes to analyze them further, so we can actually return to our work flow, and then we didn't save it actually, so we have to add it again. So we would add this analyze filter table and connect input to here, and then we will go to the workflow and we'll put here this condition. So all the rows which expression is true will be saved in this table. But actually, let's create a table name which we want to see as a filtered. So basically, this will be the up-regulated genes, transcriptomics up. So this is a table, and the name we take from lemma transcriptomics, and we add up. Okay, and then we connect this to this. Sorry, okay. So the same way we now will add also for down-regulated genes and for the methylation array as well. So let me just pause. So you can see we have add now the and the filtering of for the down-regulated genes, and now for the up-regulated CG methylation, and also for down-methylated, I would say, up-methylated and down-methylated methylation accession numbers. So the next, now we have those accession numbers, and now we have to, and here we have the respective genes. So the next would be to actually combine the transcriptomics and metylomics data, DNA methylation data together. So as we described before, we want to select promoters of these upregulated and downregulated genes and check actually which genes contain in the promoters the methylation points, differentially methylated CG nucleotides. So for that we have to convert these gene lists and the accession number list into the genome coordinates, so-called tracks. So there is a function here which convert table into the tracks. Convert actually track to gene set, but we need a gene set to track. So it will take a gene set and create a track, so the set of genomic coordinates for the promoters of the genes listed here in this file. So we connect this to this. And then basically we have to specify here what promoter regions we want to consider. Let's consider 2000 nucleotide upstream of start of transcription and 100 nucleotide downstream. And we will have to save this as a new file which will contain this track information. So we will write it transcriptomics up-regulated gene track, and this is a data element, and it will take the name from this transcriptomics up, and we add here track. Okay, and we connect it here to here. As for the methylation, before converting it to the track, we have to know those coordinates for the accession numbers. And for that, we have to annotate this table with accession numbers. Using these accession numbers, we have to annotate with the coordinates. So we have a file which contains those coordinates, and this file is located here in the public section of the geneXplain platform. So we have here for the DNA methylation this table which contains the coordinates in this map info for each of the CG accession number. So we have to annotate our table with this information about the chromosome and the position in the chromosome. The only thing you have to take into account that this is actually the location, the coordinates for the human genome build HG19. So next we will have to convert it to HG38. But let's first annotate it by putting here the annotate table function. So it will take this table So we then we take the output into the new file, which we call this will be a CG up positions in HG19. So this will be the data element, and we take the name from CG up, and we just add here post HG19. Okay, so we'll connect this to this. Next will be actually to convert these positions into the into 90 in 38, the build 38. So for that we will use another method which is present here in the Galaxy, which is liftover method. So we put it here from Galaxy directly. So, and this method will lift over the coordinates from HG19 into HG38. So we have to specify this conversion direction. So we want to convert HG19 into HG38, and we of course have to specify now the output tables. So this will be the table CG up positions HG38 and data element, and it will take name from again CG up, and here we add position HG38. Okay, so then we connect the coordinates, but we have to create also a file for unmapped coordinates. CG up post unmapped, and data element, so the same, we will say unmapped. Okay, so and then we connect this one here. So the same way we will do it for the down-regulated genes and down-methylated CG dinucleotides. So then we specify all the gene to set to track for the down-regulated genes now, and also conversion of the methylation to the positions, and also conversion them into the new build. One thing which we forget is actually in this annotation table we have to add the source of annotation. So as we know, it is actually in this table which is prepared already and in the public repository. So here we had a look. So we have to add this table to this folder. So actually we need to simply to add this URL of that table here into this folder. No, it doesn't work. Okay, so we have to do it a bit more complicated. So we have to specify this table here. Right, and then we have to connect this table to the annotation source here. And the same is for the down-regulated genes, we have to connect it to the same annotation source. Annotation source here. Alright, and then we have to select which annotation we want to add. We want to add chromosome and map info. And here, actually, we have to do it again. So I remove this H. So we have to choose the annotation source, connect this table, and then specify chromosome and map info. So now we're ready for the annotation. Let's save this workflow. Next, in this workflow, we want to find those methylation positions which are differentially methylated, either up methylated or down methylated, in the promoters of upregulated or downregulated genes. Basically, we have 4 combinations. So, because we don't know whether the methylation affect upregulation or downregulation, we can combine all these in pairs. So, actually, we have to filter the methylation points, positions of the methylation points across the promoter positions of the promoters of respective differentially expressed genes. So we have this function which is filter one track by another. So, and let's specify the following conditions. So we, for instance, we take the up methylated positions and intersect them with up methylated genes. Right? We can also do by filtering, selecting up methylated positions and filter them. Sorry, that has to be here. So up methylated positions. Filter by down-regulated gene promoters. So, and we have to specify the output track. So the output track will be those CG methylated positions which were up methylated, but they were actually filtered by down or upregulated genes, for upregulated genes. So actually we select the data element. So let's select this methylated positions up, and then we add here upregulated genes. So let's have a look. So upregulated, so we have to connect the output of this function to this table. So the same way we will do it for all 4 combinations. Let me do it. So we created now 4 entries for the CG dinucleotide up methylated, filtered by upregulated genes, up methylated filtered by downregulated genes, down methylated by upregulated, and down methylated by downregulated. So 4 combinations. Now each of these positions of the CG methylation dinucleotides we want to analyze with positional weight matrices to find the overrepresentation and enrichment of transcription factor binding sites in the regions around those differentially methylated dinucleotides. So to do that, we have to extend these positions into the areas, adding some regions left and right around those positions. To do that, we have these new methods which can do that. So we basically take a method called modify, or just let me check. We use a method called process tracked with sites. So actually we will add here this track. and we will tell the system which extension should be given to the upstream and downstream of each position of the methylation nucleotide. So let's add, for instance, 250 nucleotides upstream and 250 downstream. So we have in total 500 nucleotides to analyze transcription factor binding sites. We want, if this position of methylation are located nearby, we want to merge them if they are overlapping. And so basically that's it. So now we want to create an output track, which will be the extended track. So we get CG up, filtered by upregulated genes, and this will be 500 nucleotide extended. So it's a data element. We give it a name up, but we add here to the name 500. So it has this new name, and we connect this to this. So we of course have to specify here some more parameters. So we have to specify which genome build we are going to use here. And we simply can select this genome build from the databases. So we want to use this human genome, the latest build, which is 91. And we use this chromosome 38 build. So it is specified here. We don't have to shrink it. So it's ready to analyze. So we basically then we'll do the same for the other tracks. So let me do it. Okay, so we add all 4 tracks. Now the workflow is ready, so we can see it in. So it's pretty big workflow. So it starts from the initial data. It will generate actually 4 tracks of the positions for analysis of binding sites for transcription factor binding. So finally, after reviewing and editing of the workflow, here the final version of the workflow. So the user puts here the transcriptomics and DNA methylation data. Then there were several conversion and lemma analysis. We have added a little bit here from annotation of the table. We have to convert this table first into the track, and then this track is then converted from HG19 to HG38. So this step had to be added to the original workflow, which as we have seen it before. So finally it is ready. So we also added here the species sequence selection as a separate item in the workflow, so to make it really running smoothly. So at the end we got this workflow will create 4 files corresponding to the tracks of the CG methylation positions with 500 nucleotides around it, and these tracks are going to be used later for analysis of transcription factor binding sites. So when this workflow is ready, so we can basically start it. So for that we click on this button, so to initiate this workflow. Now the runtime environment is created for this workflow. So and it will be ready to start and to put the data in the initial parameters, and then the run workflow as we have seen it before at the beginning of the small example. So now this start page is ready. So what we put here is transcriptomic data. Let me put it here, and DNA methylation data, and we select here the tumor samples, these 2 tumor samples versus control samples for the transcriptomics, and also select here tumor tumor samples versus control samples for the DNA methylation data. So the workflow is ready to start, so we simply click the button Run Workflow. So it's now initializing to start the run. And the workflow run will take around 5 to 10 minutes to analyze all this data and to prepare the files. So we'll look after it is finished. So workflow has just finished. You see it's completed. So and it generated several files and folders. So the final files are these four. So these are actually, if you click on them, if you click on one of them, it will open a genome browser and it will show these intervals, these MISC features called which correspond to the methylation sites which are differentially methylated in the promoters of differentially expressed genes. So we can also put here the actually the positions of the methylation dinucleotides. So they are here. We can make it like this. You see that's, for instance, the methylation position, and we took 250 nucleotides upstream and downstream from it to have a region to analyze it with a genome with transcription factor binding sites. And this is actually for the down-regulated genes, and these are down methylated positions. And for example, these are up-regulated genes and up-regulated methylated positions. So we can see them in a different genes, of course. So and we see those up methylated dinucleotides. So in this case, this gene, and it's extending 5' area of this gene, and in that area, this particular region will be analyzed for transcription factor binding sites in the next workflow. So that's how we created and run the workflow for the analysis of transcriptomics and DNA methylation data together. Thank you for watching.